Yes, good afternoon, guys. Once again, welcome to another lesson. And previously, we talked about mitosis, where we saw the phases, the stages, the events, the significances, and where mitosis occurs. And like how you've reviewed the lesson, they should really be part of you. Okay? They should really be part of you. Because I'll set a question like how we've been discussing. I'll give you a cell and I say identify which phase is this. I'll say describe the process of maybe meiosis in cells. So if you've not been sober enough, then it might give you a bloody nose. But if you've been sober enough, then it becomes a walkover. So feel free, get your pens, get your notebooks. If you have a question, feel free to raise your arm. I'll see you and I pick you up and you will ask all your questions. Okay? Yes. So like we discussed, we saw that cell division is a process by which a cell divides to give rise to daughter cells. And like I always say, I don't know why daughter cells, not son cells. You begin with one cell and the cell divides to give rise to daughter cells. So these are the daughter cells. Now, we saw that it has two types. Well, the first one we agreed it was mitosis, and the second one is meiosis. And we saw what mitosis is and how it occurs. Now today we are going to see what meiosis is and how it occurs. Now meiosis is a type of cell division which involves reduction in the number and the amount of genetic material. And one cell divides, I think it's the next stage. Okay, no. One cell divides to give rise to 4 daughter cells. And each of the 4 daughter cells has half the genetic material as compared to the parent cell. I told you genetic material refers to DNA. Okay? If you've been with us right from the beginning, you know what DNA is. You know that the DNA is the one which stores all the information. And I usually say that actually you are what you are because of your DNA. So the same applies to this. Now with meiosis, you're going to divide one cell, this is the parent cell, with its complete DNA, and we usually represent it as 2N, or you can call it diploid or diploid, depending on your kindergarten. And then this cell divides, okay, actually it undergoes 2 divisions, 2 meiotic divisions to give rise to 4 daughter cells, 1, 2, 3, and then 4. To give rise to 4 daughter cells. And each of the daughter cells formed is going to have half the genetic material. It is usually represented as N, and this N is known as haploid. So this is meiosis. And it takes place, it occurs in reproductive cells, okay, during formation of reproductive cells. And when I say reproductive cells, I mean the sperm and the ovum. Now, as a good student, in your mind, you should be asking yourself that why all this? Why should you divide by meiosis, not mitosis? And like I've been saying, in genetics, you know, these sperms, they undergo fertilization, or during copulation, a male is going to release a sperm which will swim up to the oviduct, up to the fallopian tube, and they meet this ovum or the egg, and the two will fuse with each other. So if this is the ovum, and then this is the sperm. Surprisingly, the ovum is the largest cell in the largest cell, and the sperm is the smallest cell. You can imagine. But the two, when they come, the sperm is going to cross the ovum and then releases its head, and the head will fuse. Actually, the head contains the nucleus, which will fuse with the nucleus of the ovum. And when that occurs, then we shall say that fertilization has occurred. Now, since it involves fusing of the two cells, then there must be a need of having the cells, so that when you combine half with another half, then you form a complete cell. That's why they say when a man meets his woman, they form one. I don't know which mathematics is that, but this is how we try to explain that mathematics. But now imagine if you are forming such complete cells. The cell is complete, and a complete cell is also fusing with a complete cell. You produce a cell with double than what is required. Now, what if this one also fuses with another one that has double? Then it will just be confusion. Confusion will be carried from generation to generation to generation. And actually, it will be amplified. But to avoid that, then that's when we shall fuse. We shall divide these cells by meiosis. We form half, and then also the ovum, it is also half, so that when the two cells fuse again, they'll form a complete organism. And so by that, you'll be able to pass it on to the next generation, next generation, next generation, and you avoid such errors. So like we said, meiosis occurs in two different divisions. Meiosis is very similar to mitosis because they all undergo the same phases. The phases are the same, and the events are kinda the same. Actually, for meiosis too, like how we are going to see, it's exactly the same as mitosis. But the major difference is that with meiosis, for it you undergo two successive divisions. What do I mean by successive divisions? When one division occurs, it is immediately followed by another division. Okay? So those are the two successive divisions. And these divisions are meiosis 1, that is the first meiosis, and it is actually this that is so good, that is really interesting. Okay? That you need to take a good care of it. And then after you've divided in meiosis 1 and you form actually haploid cells, then you need to separate the chromosomes so that you form your 4 daughter cells, as we are going to see. So just keep calm, okay? Just keep calm. You will have fun. You will enjoy the science. Now, this is where the other important terms are really going to be important from. And before I say anything, let me first remind you what homologous chromosomes are. Now, if this is my chromosome, okay, if this is my chromosome, and my chromosome divides, not dividing, but replicates or duplicates or doubles, depending on which word is easy for you. So my chromosome replicates, maybe this is chromosome A, to give another chromosome A. Not so, guys. In case of any question, please raise your arm and ask. So, oh, sorry for that. Sorry for that. Okay, let me wrap it. Yeah. It's okay. Yes, this is chromosome A and another chromosome A. Okay? And another chromosome A. These two chromosomes are the same. They are the same, they have the same information, everything is the same. So because of that, then they are referred to as homologous chromosomes. Homologous chromosomes. Also, yes. Now, these two chromatids, this one and this one, they are referred to as sister chromatids. They are sissies, they are sisters because they are chromatids of the same chromosome. It is going to be important as far as this work is concerned. And then this one, are you seeing? And this one, they are chromatids of different chromosomes. So they are referred to as non-sister chromatids. So guys, but of homologous chromosomes. They are homologous. Homologous means the same. Now, I hope this one is okay. Now, let's talk about meiosis 1. Now, in meiosis 1, remember in interphase, you would have divided the DNA and the chromosomes would have replicated. Now, in meiosis 1, the key feature here is to form the two homologous chromosomes. They pair up, okay? And that process is known as synapsis, and form a pair of chromosomes. And then, after they have formed a pair, the two homologous chromosomes will migrate up. They will arrange themselves on the spindle equator and separate and migrate to opposite poles. That one is key. That one is really key. So this is it. You have this chromosome and you also have another chromosome. Now these two chromosomes, they will arrange on the spindle equator, say that when this one pulls towards this side and this one also pulls towards this side, they will separate and this one moves towards this side and this chromosome also moves towards that side. So meiosis 1 involves separation of homologous chromosomes. Now with meiosis 2, the two chromosomes that are now at opposite poles, they are still chromosomes, but we are interested in chromatids. The two now chromosomes, or the two chromatids, the sister chromatids of these homologous chromosomes, they will have to separate. We are going to see all this, okay? We are going to see all this, okay? So here you pull this, and you take it. Why is my pen misbehaving? You take it this side, and you also pull this, and you take it that side. So that now each of the 4 daughter cells formed. Because when you pull this one this side and you pull this one this side, you form 2 cells. Each of the 4 daughter cells will now therefore have a representative from this chromatid or from this chromosome. And so as a result, you'll be able to form cells that will carry your genes to the next generation. So basically, this statement summarizes all that we have talked about. In the first meiotic division, it involves separation of homologous chromosomes. Okay? That is the key. If you understand this, if you really understand this, then trust me, this meiosis is going to be so easy for you. The first meiotic division involves separation of homologous chromosomes, whereas the second meiotic division involves separation of the other sister chromatids. And that's all that is going to happen throughout the entire process, what we are going to discuss. And the next step over is, I guess, the phases. Okay, where we shall start with the prophase. Yes, after prophase, which other phase? Yes. From prophase, then you go to metaphase, and then from metaphase, which other phase? Yes, anaphase, and then from anaphase, which other phase? Yes, telophase. And so that's basically what we are going to talk about. But now for this one, we shall talk about meiosis 1, and in meiosis 1, we shall see prophase 1, metaphase 1, anaphase 1, and telophase 1. That is if it exists. Yeah, if a cell undergoes it. And then after all this, we talk about meiosis 2, where we shall be able to talk about prophase 2, metaphase 2, anaphase 2, and finally telophase 2. But the good thing about this is that meiosis 2 is exactly the same as mitosis. So if you really understood mitosis, then this one is going to simply be a walkover. Can I receive some questions?